Hello everyone. This week we're going to talk about parasitology. We're going to discuss some different protozoa. Some of them are pathogenic, some of them are not, so let's go ahead and get started. So what you can see right here is a family tree for the protozoa. There are different phyla. Now you will have to know the different names of those phyla, and you will have to know the organisms that are the microorganisms that are found under each phylum. So for example, we have a phylum called Ciliata, and those are called Ciliata because usually these microorganisms, such as Paramecium, and Bellentidium coli, they will usually have cilia that would help them move. And then we have phylum Amphizoa. Example, we have Entamoeba histolytica, Amoeba proteus. And then we have phylum Archezoa. We have phylum Euglenozoa. And this is where you find the euglena, which is actually a photosynthetic protozoa. And then we have Plasmodium under the phylum Apicomplexa, and this is the one that can cause malaria. So we're gonna, discuss these microorganisms right there, but again, you will have to know the name of the phylum, and you will have to know also the name of the microorganisms. The spelling will count. And you will also have to identify these microorganisms if you are given a picture. So let's go ahead and start with the first phylum, so phylum Ciliata. And as I said, microorganisms under this phylum, they're going to move using cilia, which are basically tiny hair-like structures that are going to sway back and forth, helping the microorganism move. One microorganism that falls under this phylum is Bellentidium coli, and it kind of looks something like this under the microscope. Again, be able to identify it if you are given such picture. And so it does kind of have like this bean-like shape to it. The nucleus, okay, so its nucleus kind of looks like a bean shape. So this whole entire thing is the Bellentidium coli, and what you're seeing in there, this is the nucleus, and it kind of has a pretty unique shape to it. Kind of looks like a bean. So this is kind of what can help you identify whether this is Bellentidium coli or not. So look for that bean, or sometimes it's like a bar shape. So again, sometimes it's not, very distinctive in terms of like that bean shape. So if it's kind of something like this, kind of like a bar shaped, again, this is still considered, Bellentidium coli. Usually what happens is the cysts, those kind of like dormant stages of these protozoa, they can get ingested by us humans if we drink contaminated water, or if we eat contaminated food that has those cysts. Then we would ingest those cysts, and then it will start developing, and then you would have Bellentidium coli inside of you. When someone is infected with Bellentidium coli, they're going to have diarrhea. So one of the symptoms that they're going to have is diarrhea. Another one that we're going to talk about that also falls under the phylum Ciliata is Paramecium. SPP meaning different species. So Paramecium, we have so many species of Paramecium out there. Now Paramecium, those are free living microorganisms, meaning that they don't need a host, and so therefore free living microorganisms that don't need a host, you would know that they are not pathogenic. They're not going to cause disease. This is how the Paramecium looks like. Now, what you're seeing right here are two different types of reproduction. When you see the Paramecium next to each other like this, then they are going through sexual reproduction or conjugation. When you're seeing the Paramecium kind of facing each other like that, that is asexual reproduction, where basically one of them goes ahead and kind of makes an identical copy of itself. So this is basically binary fission. So this is asexual reproduction, how it would look like under the microscope. And then this is sexual reproduction. And you can see, by the way, those tiny hair-like structures right here. You can see it better on this image versus that one. And those are the cilia. Those are, those hair-like structures that's going to help the paramecium move. Now we're going to move on to another phylum, phylum Amoebozoa, and microorganisms under this phylum, they move using pseudopodia. Now pseudo meaning fake, podia meaning feet, so we refer to it as fake feet. Now this example right here of the microorganism that we're going to talk about, Entamoeba histolytica, you can't really see the pseudopodia, okay? So they are invisible and you can't really see them when we are looking at an Entamoeba histolytica. And they have this like oval shape to them. And their nuclei kind of looks like a ring with dots inside. So right here, here's the nucleus, and it kind of looks like, it has a dot inside of it. So you can see the nucleus right here. You can see the nucleus right there. So again, this is how Entamoeba histolytica would look like. This is a pathogenic protozoa, and again, one of the major symptoms that someone would have if they have Entamoeba histolytica infection is diarrhea. And again, pseudopodia, those kind of fake feet are not going to be visible. Now another one under the same phylum is Amoeba proteus, and Amoeba proteus kind of looks something like this. So this is how they would look like under the microscope. Again, a pretty unique shape, kind of looks like stars, right? This one looks like a star. Amoeba proteus, you can see the pseudopods. So these extensions right here, okay, these extensions that you're seeing right there, those are the pseudopods. So definitely the pseudopods for the Amoeba proteus, they are visible, and those are free living. So again, no need for a host, and so therefore, they're not going to cause disease. Now we move on to another phylum, Archaeozoa, and microorganisms under this phylum, they usually move using flagella. And so again, those flagella kind of like whip-like structures. You can definitely see the flagella right there. You can see the flagella right here. And so they're pretty visible, right? So this one right here is the Trichomonas vaginalis. And this is how it would look like under the microscope. So they're kind of like round, right? Big round microorganisms. And you can definitely see the nucleus, right? It's pretty visible, right? That kind of dark blue dot in there, that's definitely the nucleus. This large one right here, that's the nucleus. And you can see the flagella kind of hanging on one end. And so this is how, again, they would look like under the microscope, and it would cause a vaginitis, so kind of like inflammation of the, vagina, and definitely those are pathogenic, okay, so they are going to cause, an infection, and they're going to lead to symptoms. Another one under the phylum Archaeozoa, we're going to talk about Giardia lamblia, and this is how they look like. They have a pretty unique shape to them, kind of like a teardrop, and it kind of seems like they have eyes. Right, so kind of like looks like, they have eyes right there, and these are their flagella, kind of like, flying everywhere. So this is how they would look like, and again, it's a pretty unique shape, so it's kind of easy to spot them and know that you're looking at a Giardia lamblia. And so, as I said, they have this like pretty unique teardrop shape with those two nuclei that kind of look like eyes, and then they have multiple, flagella hanging out of them. or hanging off of them. They are pathogenic, so they are going to cause an infection, and the main symptom that someone would have would be diarrhea. Then we move on to the phylum Euglenozoa, and those move also using flagella. But when you come and take a look at them under the microscope, you can't really see these whip-like structures, so they are not visible. An example, by the way, this is a blood sample, so what you're seeing right here are the red blood cells. So these, they would go ahead and inhabit the bloodstream, and those are called Trypanosoma brucei, and they kind of look like an S, okay, kind of like squiggly shaped really, and as I said, you would find them in the bloodstream, and those cause a neurological condition called the African sleeping sickness, and the reason why they call it the sleeping sickness, because whoever has it would feel very, very tired all the time, and they would sleep a whole lot, so they would kind of feel like very sleepy. So that's why they called it the sleeping sickness. Another one under the phylum Euglenozoa, we're going to talk about Euglena, and there are so many different species of Euglena, by the way, and this is how they would look like under the microscope. Again, they are able to move using flagella, it's just you can't really see the flagella under the microscope. They kind of look something like this, thin like structures over here, they're stained, that's why you kind of see them a pinkish in color, but in reality they're actually greenish in color, and those are free living, so they're not going to cause disease. They are actually photosynthetic, so they can make their own food. Then we move on to another phylum, Apicomplexa. Those are non-modal, so they can't move. So they look something like this. This is an example of Plasmodium. And again, there's so many different species of Plasmodium. And by the way, again, this is a sample of the blood, and those are the red blood cells. So you see that the plasmodium actually enters into the red blood cell itself. So not just, they're not just found in the bloodstream, they actually penetrate all the way into the red blood cell. So here's the red blood cell, and you can see that within this one red blood cell, you have one, two, you have three plasmodium in there. And so they, again, they are going to be found inside of the red blood cells, and they kind of look round like this. Kind of looks like a ring, right? So with that nucleus on the edge, so they kind of look like rings. And as I said, they penetrate deep into the red blood cell, and those can cause malaria. Let's move on to another group, right? So we're going to move from protozoa, and we're going to start talking about fungi. So again, we have, kingdom fungi, and under this kingdom, we have two main groups that we usually discuss. The yeast, those are the unicellular, fungi, and then molds, those are the multicellular fungi. And under molds, we have, three main groups that we talk about. Penicillium, and this is the one that they extracted the penicillin antibiotic from it. And then we have Aspergillus, and then we have another one, which is Rhizopus. So let's go ahead and kind of, take a look in more detail and talk about fungi. So as I said, yeast, they are known to be unicellular, so they're only made up of one cell, and they're kind of like, round or ovalish in shape. The molds, those are the multicellular. And so when you have a sabdex plate, for example, and you see all of that hairy, fuzzy growth on it, that would be mold. And that fuzzy, hairy, like, texture or, growth, basically, that you would see on the sabdex plate, it would be a bunch of hyphae. So those are the hyphae, and when you have a bunch of them together, we refer to it as mycelia. So again, those are the hyphae, and when they're grouped together, we refer to it as mycelia, or singular, that would be a mycelium. Now, fungi, they produce spores, and again, those spores for the fungus would be a reproductive spore. Now, here would be like, here's the hyphae, here's an image of the hyphae, and then at the end of the hyphae, you can have the spores. Now, if the spores are enclosed within a sac, we refer to it as sporangium. And once that sporangium, kind of like that sac, opens up and bursts open, all the spores are going to be released. And as I said, those spores are used for reproduction. Now, this is an example of fungi out there that their spores are not really contained within a sac. So they have like their spores like this, naked, not, they're basically exposed. This is how penicillium would look like. Again, make sure that you are going to be able to identify if you're given images. So this is how penicillium would look like. And then this is another pretty unique one, how it looks like under the microscope. This is called aspergillus. So this one is aspergillus, this one is penicillium. And again, their spores are not really enclosed, right? So they're not enclosed inside of a sac, and so we call their spores conidia. And those conidia definitely would look different from one fungus to another. But go ahead and, as I said, make sure that you are able to differentiate and identify them under the microscope. We also have another example. This is the rhizopus fungus, and you can see that their spores are actually, found within a sac, and so we call it the sporangium. And so the spores would be found within this sac right there. We move on to another group, and those are the helminths, and those are basically the worms. So when we talk about worms or helminths, there are two main groups that we discuss, the flat ones, the flatworms and the roundworms. The flatworms are the platyhelminths, the round are the nematodes. And those are basically like examples, right? So this is an example of the nematode. And then if we take a look at the flatworms or the platyhelminths, there are two subgroups. There are the flukes and the tapeworms, or basically trematodes and cystodes. And again, this is right here, just gonna showing you examples. Make sure that you do know the names, as I said, of the phyla. Know what's the other name for a flatworm. So the phylum would be platyhelminths. And then know again, for these subgroups, tapeworms would be the cystodes, flukes would be the trematodes. Now flukes, under the microscope, they look like a flat body, and I'm going to show you an image. So they're not segmented because they're flat. On the other hand, tapeworms look like they are actually made up of blocks connected to each other. So under the microscope, you're going to see that they are segmented. Now, when we come and talk about the sexual status of these worms, there are two sexual statuses that, we could talk like that we're going to discuss, monoecious and dioecious. So mono, meaning one. So in one worm, there are actually both the male and the female reproductive parts within that one worm. Di coming from, two, right? Di meaning two. So dioecious, you have two separate worms. One is a male and the other one is a female. Versus with the mono, it's just one worm that actually has both male and female reproductive parts. And again, this is just kind of showing you examples of which ones are considered mono, which ones are considered di. This is how a liver flukes. Remember how I said the liver, flukes in general, they look flat and they don't have any segments. So this is how a liver fluke would look like. The liver fluke, by the way, is like its common name. This is the scientific name. And they are under the group of platyhelminths, those flat worms, and they are under the subgroup called trematodes. Another, platyhelminths that's under also the group of trematodes, the Schistoma mendelssohni, and it looks something like that. Now, there are actually, a female worm and there is a male worm. So those two worms are definitely, separated from each other. So there's, a female and it's going to have the female reproductive parts, and then there's the male and it's going to have the male reproductive parts. This is Tenia pisiformis, and this is under the group of cystodes. And again, it's under the main group known as the platyhelminths. And so this is how it looks like. See how the body is segmented? And that's what I meant, earlier when I said some are segmented and some are not. So these are segmented. You can see these segmented parts right there, and within it you can see that this worm right there has both the testes and the ovaries. So this one is definitely a monoecious worm, and this is how the head looks like. And usually right here at the very edge of the head they have these hooks. And so they use the hooks so that they can grab on to the host tissue. This is, nematodes, and this is an example of Trichinella spiralis, and these worms would actually bore themselves into someone's skin, so they actually have the way to make it through someone's skin, and then in there they're going to start producing the cysts, which are kind of like the dormant stage, and they can go ahead and start multiplying inside of a host tissue, and they usually bore themselves and they curl themselves into a muscle tissue.